Everyone. A very good evening. Today I'm going to talk something about biosafety regulations in India. What is this biosafety and what are the regulations which are governing it for the safety? So, coming to biosafety, by definition, it means the need to protect the human and animal health and the environment from the possible adverse effects of the products of modern biotechnology. That's defined as biosafety. Today in this class we are going to study something like biosafety levels. What are all the legislations that are governing this biosafety in India, and something about transgenics and what are the safety measures. Now, coming to the basic thing, the types of biosafety levels. So there are four types of biosafety levels according to the risk factors involved, depending upon the nature of the pathogen that's being handled. So there are biosafety level 1, where it is meant only for basic teaching and research. So normally the biotechnology labs in the colleges for academic teaching and some kind of basic research. Uh, these are all coming under biosafety level 1. Then biosafety level 2, the primary health services, the diagnostic research services and research labs comes under BSL2. Then BSL3, the basic biosafety level 3, special diagnostic service and research. And finally biosafety level 4, that deals with the dangerous pathogen units. So in this way it's classified. So the level of safety precautions increases from biosafety level 1 to level 4, and also at the same time the danger uh, increases from biosafety level 1 to level 4. So coming to this biosafety level 1, laboratory type is meant for basic teaching and research, and laboratory practices followed in this are good microbiological techniques are followed in this laboratory, nothing more. And safety equipment, it has nothing like safety equipment, and open bench work is allowed. Then coming to basic biosafety level 2, the primary health services, diagnostic services, research and all are all dealt here. And the laboratory practices along with good microbiological techniques, these are compulsory things. Along with those, protective clothing, biohazardous signs on the laboratory are being imprinted. Then safety equipment, so open bunch here also, along with the biosafety cabinets for aerosols. Aerosols are airborne, which may be hazardous when inhaled. Right. Then coming to biosafety level 3, so containment biosafety level 3 here. So the previous ones are basic biosafety level 1, basic biosafety level 2. The third one is containment biosafety level 3. So laboratory type is meant for special diagnostic services and research. Here, along with all those things which are present in level 2, special clothing, access control, only it is accessible to few, and then directed airflow is happening in this containment biosafety level 3. Then what are the safety equipment here? The biosafety cabinet, and along with that other primary devices for all activities. What are these primary devices and all we will learn. Then maximum containment biosafety level 4. This deals with dangerous pathogen units. Here the level 3 along with airlock entry. Then shower exit. So when you are coming into and moving out of the lab, it is airlock protected. Then you have a shower exit. So after completion of your work, you need to take a shower. After then exit. So there are also protective decontaminants used in the shower, and also special waste disposal facilities are associated with this laboratory. Then here the safety equipment are class 3. So the biosafety cabinets are class 1, class 2 and class 3. So here class 3 biosafety cabinets are positive pressure suits, so which I have shown in the opening slide. Uh, Normally, you people might be looking in regular TV kind of activities where people are wearing something like a spacesuit and working in the laboratories, especially related to the present COVID-19 problem, with class 2 biosafety cabinets and also double-ended autoclaves. So these are the basic differences associated with the biosafety levels, type of work involved, what are the laboratory practices followed, and also the safety equipment they are contained. Then what is this biocontainment? So the principle of holding or being capable of holding or including within a fixed limit or an area. So it is confined. So the preventing the unintentional release of the biological agents through a combination of laboratory practices, uh, where containment equipment, that is a primary barrier. So containment equipment is referred to as a primary barrier. So your biosafety cabinet is some kind of primary barrier. Then, and also laboratory facility design. So laboratory facility design, how the rooms are there, how it is away from the uh, main building, or it is within the building. So normally those biosafety level 3 and 4s are normally away from the main buildings. They are separate laboratories. Then Convention on Biodiversity 1992. CBD, this is a Convention on Biological Diversity. This is also called as CBD. It was the year is 1992. The focus is conservation and sustainable use of biodiversity. It has recognized the potential of modern biotechnology for human well-being. It took the cognizance that the modern biotechnology could have serious effects on environment and health. Definitely, there are two sides of every coin. So modern biotechnology, though it has its positive effects, it has its negative effects on environment and health as well. So these to be stopped from entering into the ecosystems. So Article 8G emphasized the need to regulate the risks associated with the use of LMOs, living modified organisms. And then Article 19.3 set the stage for legally binding international instrument about biosafety. So all these biosafety guidelines have to be followed definitely by all those people working across the country in various labs, in various institutions, have to be legally bound with these biosafety regulations. Then Cartagena Protocol on Biosafety. So it has entered into force in 29th of December 1993. The focus was on transboundary movement of these LMOs, living modified organisms. So transboundary movement in the sense moving across the country. So seeks to lay down an internationally acceptable framework to provide for an adequate level of protection against these possible adverse effects of LMOs on the biodiversity and the human health. So there are a lot of movies, if you observe the Terminator series or some kind of Hollywood movies where LMOs, living modified organisms, which are created in the lab, move into the ecosystems and disturbing, creating some kind of disturbance. Then basic premise of the transgenic crops should be substantially equivalent to their conventional counterparts and pose no greater risk. So this is the main motto, basic premise of the transgenic crop. So transgenics, you know much better. It has a gene insert from some other organism into it, which has conferred some kind of advantage, additional advantage to it. So it should be almost so soybean, which is transgenic. It should be substantially equivalent to its conventional counterpart. So normally non-transgenic and transgenic should be almost alike. There should not be any kind of nonsense or unnecessary thing associated with it, except what is the advantage, what we want to have out of a transgenic. That is more than that, than having some additional risk associated with the transgenic. So that's a main basic premise. Then Codex Alimentarius Commission. So it was established in 1962 by WHO and also FAO. So Codex is a collection of internationally adopted food standards that protect the consumer's health and ensures the fair practices in the food trade. So the task force on biotechnology derived foods has been formed by this Codex Alimentarius Commission. Then genetically modified organisms and rDNA products are governed by some kind of rules and regulations. Uh, they are like this Environmental Protection Act 1986 and rules 1989, Industries Development and Regulation Act 1951, New Industrial Procedures, EXIM policy, then Drugs and Cosmetics Act 1940, Rules 1945, Pharmaceutical Policy 2002, Seed Act 1966, Seed Rules 68, Seed Control Order 1983, PVP, Plant Varietal Protection and Farmers' Right Act 2001, National Seed Policy 2002, Plant Quarantine Order 2003, Food Safety and Standards Act 2006, and so on. Number of things. All these things are governing our genetically modified organisms and rDNA products. Then the Indian Environmental Protection Act is very important. EPA 1986. It's provide protection and improvement of the environment. Environment includes what you know, air, water, and land, and their interrelationships which exist among and between the water, air, and land, and humans and other living creatures, plants, microorganisms, etc. That's all. Environment, what we call. So the statutory authorities include Recombinant DNA Advisory Committee (RDAC), Review Committee on Genetic Manipulation (RCGM), Institutional Biosafety Committee (IBSC), and Genetically Engineering Approval Committee (GEAC), State Biotechnology Coordination Committee (SBCC), and District Level Committee (DLC). So these are the statutory authorities which are under genetically modified organisms regulation. So the regulatory procedure, how it follows, you observe Institutional Biosafety Committee, that's forwarding applications for approval of RCGM. Then RCGM receives the application, Review Committee on Genetic Manipulation. So where greenhouse experiments, contained field trials, that is in-house trials, initial hybrid trials, generation of the data on gene stability and expression and confirmation of the gene or event, etc. All these things happen with these kind of thing. Then RCGM, then approval for conduct of multi-location field trials on the selected varieties or hybrids. Then followed by that, monitoring cum evaluation committees through state agriculture universities. Here evaluation of multi-location field trial data and recommending to GEAC under intimation to RCGM on suitable variety or hybrids for large scale trials. Then coming to GEAC, so seed production for 10 hectares, then large scale trials, seed production up to 10 hectares, then ICR trials first year, ICR trials second year, seed production up to 100 hectares, then large scale trials and MEC evaluation of these large scale trials through SAUs, and then finally GEAC giving environmental clearance of the event or the gene in the given background. Yes, the transgene is present in the transgenic variety what we have developed or hybrid what you have developed, and the expression is stable. Yes, so for example, a BT gene is governing resistance for bollworm or Spodoptera. Then so when a transgene BT gene has been transferred, yes, that is expressing and also it is stable. So that, and the yield trials are also good. Then the material is cleared from the environmental sensitivity by Ministry of Environment and Forests or otherwise. Then if it is not, it is rejected. So this is how the regulatory procedures for the development and commercialization of the genetically modified organisms happen, especially plants. Then regulation of GMOs. You see IBSC, laboratory release, RCGM, strip trials, containment field trials, biosafety tests, multi-location testing, monitoring cum evaluation by SAUs, SAU trials, monitoring cum evaluation committees, then GEAC, large scale trial 1, monitoring cum evaluation committees, then large scale trial 2, monitoring cum and evaluation committees, then ICAR ECRIP trial 1 and 2, then finally environmental release. So the normal varieties, we call them as released as a variety. Then here we are releasing into the environment, so these are safe, they can be released. So this is what it's different from the rest normal thing. And confined field trials. So biosafety research level 1, RCGM, 1 acre of land, 20 locations, it's conducted. Biosafety research level 2, GEAC, under the GEAC, where 2.5 acres of land and variable number of locations it's been conducted. These confined field trials, so they are enclosed, they are separated from the rest. Then monitoring confined field trials, so monitoring cum evaluation and evaluation committees of RCGM. Then monitoring team consists of 3 reputed scientists, including a team leader, a plant breeder from SAU, a director of research of SAU, deputy director of agriculture, DBT or RCGM nominee. So this will be in the monitoring team. So the terms of reference, ethical conduct, so they have followed ethically, they have done nothing going wrong. Then fair presentation, so they are properly reflecting the results. They are not doing any they're not concealing anything. Due professional care, so they have all followed things professionally, they are doing the things. Then independence and evidence based approach, so these are the terms of reference. Then monitoring procedures, so preparation of the field site for visit, then documentation of inspection, everything is documented in the sense video recording, proof recording, something like that. And storage facility monitoring, then transport, storage, labeling and field trial monitoring during the growth period, termination, harvest and disposition, and post-harvest monitoring. All these things are monitored. Because this is genetically modified organism. We have to take utmost care starting from its seeding to finally disposing. Then site visit, so communication should be done in advance, understanding of the guidelines, standard operating procedures and terms and conditions of authorization, then checklists or inspection forms, copy of the field trial map, prompt and actual reporting of non-compliance or violations. So these things have to be followed. Then recording formats, so record of the transport inventory list, record of the storage, inspection, inventory, record of the planting, record of the spatial isolation, record of the harvest or termination, record of the post-harvest monitoring, record of the corrective action. So all these records have to be maintained properly. The documentation for inspection, so a letter of permit authorizing conduct of the confined field trials is the first document. Then the transport documentation, so record of the transport, then storage facility documentation, record of the storage, inspections, current season document, record of planting, spatial isolation, what you have followed, or the records for other methods of reproductive isolation, so trial, harvest or the termination documentation, then post-harvest management documentation, or any records related to compliance or corrective actions. So all these things have to be followed. Then field trial site inspection, so prior to authorization to verify the physical surroundings and circumstances of special concerns, so they have to be looked for. Then during the planting to verify the material management procedures, cleaning of the equipments used for planting, and disposition of the remaining plant material. So you have plant material in excess, you have to dispose the plant material because it should not be gone into wrong hands before testing. And during the period of crop flowering and prior to seeds, at most critical time to verify implementation of reproductive isolation. Why it's done? Because of to prevent outcrossing, because this is having a transgenic pollen. So the transgenic pollen may contaminate the rest of the varieties, and that can go in, and a gene flow may happen. Then during the harvest or trial termination to verify the cleaning of equipment, disposition of the harvested material, destruction of the residual plant material on this site. so that no rotting and no nonsense things may happen. Then during the post-harvest period, to verify if the area under the post-harvest restrictions is free of prohibited plants. So all those things have to be taken care of. Then institutional biosafety committee, it's constituted by an occupier or any person including R&D institutions handling the GMOs. So for example, Punjab Agriculture University has developed a first transgenic BD variety of cotton. So it is here I holding a institutional biosafety committee cadre. Then comprises head of the institution, the scientist doing rDNA work, medical expert, and also DBT nominee. Then assists the occupier or any person including the R&D institution prepare an emergency plan as per the guidelines of RCGM, and the copies of the emergency plan to be made available to district level committees or state biotechnology coordinating committees and district and engineering genetic engineering approval committee GACs. Then review committee on genetic manipulation. It functions in the Department of Biotechnology. What are its functions? To review the reports in all approved or ongoing projects involving high risk category and controlled field experiments, research in 4 areas, namely human and animal healthcare, agriculture, industry, and environmental management. To visit the site of the experimental facilities periodically where the projects with biohazard potential are being pursued, and also at the time prior to the commencement of the activity to ensure that adequate safety precautions or measures have been taken by the as per the guidelines. Then to issue a clearance for import or export and etiological agents, vectors, germplasm, organelle, etc. needed for experimental work, training, and research. Then evaluation of the proposals by RCGM. So they have followed rDNA recombinant DNA safety guidelines 1994. They have been updated time to time. Then genetic engineering approval committee functions under the Ministry of Environment and Forests to examine and issue the clearance from the viewpoint of environmental safety. They are not causing any harm to environment on case by case basis. So activities involving large scale use of hazardous microorganisms and recombinants in research and industrial protection from environmental angle. Then proposals related to release of the genetically engineered organisms and products into the environment, including experimental field trials. Then biosafety parameters for assessing environmental safety includes general information, rationale for the development, what is the necessity of doing this research, description of the host plant, what kind of plant it is, what is its history, or what you call them, description of the host plant, how its pollination happens, how its reproduction happens, the botany of the plant, etc. The centers of origin, diversity of the crop species, geographical distribution of the target crop, and sexually compatible plant species, including wild relatives. Then environmental parameters, gene flow, implications of outcrossing, then effect of on the target and non-target organisms, and then effect on the soil biota. Then toxicity parameters including histopathological studies, need based. So if needed only we have to go through this. So food or feed safety evaluation in animals, such as effect on small laboratory animals like rats, normally use mouse, laboratory mouse. So effect on livestock animals, representative goat studies, or are large scale animals, effect on the birds, avian species, effect on the fish. Then biosafety parameters for assessing the allergic parameters, which are also need based again. So primary skin irritation test in case of rabbits or guinea pigs, irritation to mucous membrane test in case of rabbit and guinea pig, then immunological responses in suitable animal systems, then agronomical practices, efficacy of the gene at phenotypic level. So the gene what we have inserted, is it expressing phenotypically or not? Then yield, growth and development parameters, response to major diseases and insect pests, then quality parameters and evaluation, economical evaluation, cost benefit ratio, whether it is cost effective or not. Then toxicity evaluation of transgenic seeds. So acute oral toxicity of the transgenic seeds in case of rats are used here. Then subchronic 90 days oral toxicity test along with rat. Then subchronic oral toxicity with goats up to 90 days. Then feeding studies in lactating crossbred dairy cows. then feeding studies in chicken, feeding studies in catfish, then primary skin irritation test with a rabbit, then irritation to mucous membrane test in case of especially female rabbit, then skin sensitization test of the transgenic seeds in guinea pigs. These have to be carried out as per the guidelines. Then for transgenic vegetables, acute oral toxicity test with a rat again. So subchronic 90 days oral test with rat again. Primary skin irritation test with rabbit again, and irritation to mucous membrane test again with a female rabbit. So website on by Indian biosafety rules and regulations are been kept in dbtbiosafety.nic.in, and Indian GMO Research Information System IGMORIS. This is also one more website for more details. Thank you very much, and my acknowledgements are due to Dr. P. Anand Kumar, sir. So, for using his resources in developing this PowerPoint presentation. Thank you very much. Thanks for listening.